Everyone, this is Ms. Moriarty, and I'm here to discuss with us our process known as protein synthesis. Make sure as you watch this video, you should be taking down notes, either using the follow-along notes posted to Classroom or on a separate piece of paper with a pen or pencil. Now, I know I covered this slide in the last video, but I wanted to start with this slide for this video. If you can recall, we know that RNA is kind of like the middleman, right? It's going to help to turn DNA eventually into proteins. And if you can recall, it's three types of RNA that are going to play major roles in protein synthesis and the two processes that make up protein synthesis. So let's kind of dive into our video. Remember, it's messenger RNA or mRNA that's going to actually be a complementary strand of RNA to the DNA. We know it's going to be created in the nucleus, and eventually it's going to travel out to the ribosome, into the cytoplasm of the cell, where then that message is going to be translated into proteins. And remember, big differences between DNA and RNA is the fact that RNA is single-stranded, which makes it a much smaller molecule, and so it can fit out of the nucleus. And if you can see here, here's our DNA strand. This would be an example of a gene here, since we have a sequence of DNA. And here would be our mRNA strand. If you notice, right, each base pair is going to be complementary to the one that you see here in DNA, except the only difference is we know DNA has thymines while RNA has uracil. Now remember, proteins are made up of smaller subunits or building blocks known as amino acids. And another name for a protein is actually called a peptide, and that's because of the type of bonds that they form between each other. Remember, it's individual pieces of DNA that are going to code for then eventually an amino acid. And if you can recall from our POGL and our past discussions, we know it's going to be three pieces or three sequences of DNA that creates what's called a codon. And that codon is going to then specify for one amino acid. Our mRNA is also going to have those complementary codons. So we can kind of see that here. We've got our three nucleotides. That's a part of DNA. That's cytosine, cytosine, adenine, CCA for short. Each codon is going to code for just one amino acid, and that's where we need RNA's help. Remember, we need to make a copy of that. So here, as we can see, that we've got our DNA template strand. Remember, mRNA is going to be the copy of our DNA, and it's going to be complementary to the DNA. So take a second, guys, pause the video at this point, see if you can answer the two questions that I have below. How many codons are there, and what does the other DNA strand look like? Okay, at this point, you should have paused the video and answered our two questions, so let's see if you are correct. How many codons are there? Well, actually, there are three in total. Remember, we read a codon as three DNA bases or three mRNA bases, and so we can see those are going to be grouped into three codons. Now, what does the other DNA strand look like? Because recall, DNA is a double-stranded molecule. Well, the other strand should resemble something like this. We should see that we're going to have complementary base pairs to the template strand that we see here. So it should be TGCAA, TCGG. Now it's going to be those three DNA codons that are transcribed into our mRNA codons and eventually turned into amino acids. So now we've got our DNA template strand. We have our mRNA strand that is transcribed. Now we need to do what's called translate and turn that RNA into protein language. Now we do that using what's called an amino acid chart. Sometimes you can see it viewed as a circle. Sometimes it can be in the form of a table. I'm going to walk us through how to find the amino acids for these three that you see here. So let's take a look at number one, UGC. So you'll notice up here, that's our RNA codon. Remember, it's going to be DNA. That's going to be the one that's actually coding for this amino acid. Remember, mRNA is simply just the copy. So we're going to use the RNA, and we're going to start in the middle, and you're going to find the first base is U. Then you're going to move out, and the second base we see is G. Now, the third base we see is going to be C. And so the codon UGC codes for the amino acid known as cysteine. Now, looking at number 2, AAU. Let's do the same thing. Looking at the middle of our circle, we'll start with the first base there, that's A, followed by another A. Looking at the last base, we see U. Okay, that's going to tell us asparagine. That's going to be the type of amino acid that that codon codes for. And the last one is CGG. Here we go, right? We start in the middle once again. We see our C, followed by G, and the last letter in that codon there is G. That's going to give us the amino acid known as arginine. Now, in nature, there's about 20 or so different amino acids that occur naturally, and it's those combinations of amino acids that actually gives us all the different types of proteins we find in our bodies. Now, remember, protein synthesis is broken down into 2 steps, and in the last few slides, we actually did both of these steps. Remember, to go to DNA to protein, we have to transcribe it, we make it the copy, and then we turn it into protein language. So we translate it from RNA to protein. But what is actually involved in all of that? Well, we know that mRNA is then going to be transcribed into our DNA's code. Recall, it takes place in the nucleus because that's where DNA is going to be located. And there's going to be a very special enzyme known as RNA polymerase that's going to temporarily unzip our DNA, and it's going to add complementary RNA nucleotides to then create our mRNA strand, which eventually is going to travel out of the nucleus to the cytoplasm of the cell at a ribosome. Okay, so just note, remember it's mRNA that's going to act as the messenger because it's holding the code. It has the instructions. And so now our mRNA strand has arrived to a ribosome found in the cytoplasm. And so here, this is where all 3 types of RNA are going to come into play. And this is to take that RNA and translate it into our amino acid sequence, which then makes our protein. So another type of RNA that's used in translation is known as ribosomal RNA or rRNA. This actually helps to make up the ribosome itself, and it helps to bond the amino acids together to eventually make the protein chain. Another important type of RNA involved is known as transfer RNA or tRNA. What this type of RNA does is it carries or transfers over the amino acid to the mRNA codon. And it's actually going to be tRNA that's always complementary to the mRNA strand. And you'll notice on our T-shaped molecule known as tRNA, where it's going to bind is going to be here at the mRNA. But how does it know where to go? Well, tRNA has what's called anticodons. So that's what I mean by it being complementary to mRNA. So it knows where to go because it's going to bind to those complementary mRNA codons. And eventually what it will do is drop off the amino acid that it carries and go pick up another one to then continue with the process. But just note, during translation, it is the mRNA codons that tells us what type of amino acid is going to be produced, not the tRNA anticodons. Remember, those are only there to help the tRNA find where it needs to go. mRNA is what's actually coding for the amino acid. So here's just a little closer up look. Here's our T-shaped tRNA. Here's leucine. That's the amino acid that it's going to be transferring. So here, if the mRNA codon tells us CUU, that would translate to the amino acid leucine. So our tRNA molecule is going to be complementary to that. And so it's going to carry or transfer over leucine to the ribosome at the mRNA. And it's going to know where it goes because it has the complementary anticodon, GAA. So here's what it kind of looks like in a larger reference here. Here's our ribosome. This is our mRNA sequence. You'll notice the tRNAs come in, they pair up with the complementary mRNA, and they drop off their amino acid. And over time, as they continue to do that, you'll see that we've got a very large chain of amino acids, which makes up our protein. Now, the amino acids are attached to the tRNA by, once again, another enzyme known as tRNA synthetase. So guys, take a second in your notes, pause this video and see how much of this you can label. Remember, this is our process known as translation. Okay, at this point, you should have paused the video and see if you can label our process known as translation. So number one here, guys, this is our mRNA. Notice it is a single-stranded molecule. 2 is our ribosome. So you'll see the mRNA has arrived to the ribosome. This is our amino acid. Here is our peptide or our protein being built here, followed by the tRNA that's going to be transferring each amino acid to eventually build our larger protein. Now, if we put this all together, right, we know proteins are going to be made during synthesis. Transcription occurs in our nucleus. It's going to go from DNA to RNA. Translation occurs in the ribosome, found in the cytoplasm, and the final product of that is going to be a protein. Now, some important things to take away. Remember, there's only 20 naturally occurring amino acids that we find on Earth, and there are actually 64 possible codons that can code for all of those amino acids. So more than one codon can actually specify for the same amino acid. But something really important to note, and this is actually the last slide of our video here, how does our RNA polymerase or our tRNAs and our ribosome know when to start and when to stop? Well, there are actually start and stop signals within our DNA sequences as well. And the start codon is actually always AUG. That codes for an amino acid known as methionine, and that's where it will always begin. Here are what's known as the stop codons. This is actually what's going to tell the tRNA, the mRNA, the ribosome to stop building the protein. If these 3 codons appear in the mRNA sequence, it will tell it to stop. All right, guys, that's it for our instructional video. I know this was quite a long one. Make sure as you watched, you took your notes. Write down any questions that you might have for class tomorrow, and I will go over those.